Nice. All right, so welcome everyone. My name is Emily Heald. I am the Rivers Educator with UW Extension. I work with the Water Action Volunteers Program, and we are excited to continue our Winter Water Talks webinar series with you. So this series is brought to you by the Water Action Volunteers Program at UW-Madison Extension and the Citizen Lake Monitoring Network at Extension Lakes in Stevens Point. So I'm joined by my co-hosts Katie Bradford and Paul Skowinski, who will be helping moderate the webinar today. They're going to be helping facilitate the Q&A. And we just wanted to let you know we have one more talk for our final Winter Water Talk series, and it's on February 14th on the crayfish of Wisconsin. So we'll be sending out details for that soon. I believe Katie just put it in the chat as well. And then before I introduce our speaker, I just want to share a couple tips to help you participate best in today's webinar. We are recording this for sharing in the future. I'll share a video link for people to watch later. That'll be going out to all registered attendees. And then lastly, this webinar format in Zoom might look a little bit different to you compared to webinars in the past. We don't have the usual chat box, so if you'd like to ask a question or share a comment, you can go to the Q&A tab, which is likely at the bottom of your Zoom screen, and you can submit a question or share a comment there. We're also going to be maybe pausing at a few places during the presentation with some questions. Jessica indicated she's very used to being interrupted by students with the work that she does at UW Parkside. So you can submit a question if you have any clarification questions. You can submit those, or we can take bigger questions at the end. And then you can also upvote people's questions or comments if they're of interest to you. We're going to try to respond to everyone. And without further ado, I'm very excited to announce our speaker for today. We have Jessica Orlovsky joining us. She's an associate professor in the Biological Sciences Department at the University of Wisconsin-Parkside. Dr. Orlovsky teaches a variety of courses, including invertebrate zoology, principles of ecology, freshwater ecology, and biostatistics. Dr. Orlovsky received her undergraduate degree from the University of Wisconsin-Stevens Point and a graduate degree from Iowa State University and the University of New Brunswick in Canada. Dr. Orlovsky's research focuses on invertebrate conservation and environmental monitoring. So thank you so much for joining us, Jessica. I'm going to stop sharing my screen now, and the floor is yours.
All right, let me just quickly confirm that you can see the slides okay.
It looks fantastic.
Wonderful. Well, thank you all so much for coming and for the invitation to speak and present to you and discuss some of my favorite topics. So if you've caught some of my presentations before in other venues, some parts of this story might be a little bit familiar, but this is a new way to put everything together in one place. And we're really going to focus on how invertebrates can tell a story of their stream or their habitat. So I know this is kind of a difficult format to be interactive. So I'm going to ask you to use your imagination for a moment. And maybe if you've got, a piece of scrap paper next to you, you can write things down. But I just want to get you to quickly evaluate. And this happens every time my slides don't want to advance. There we go. Compare these critters. So if you've been involved with aquatic biomonitoring, maybe you've encountered these. If you haven't, that's fine. If you don't know their names, even better. What I want you to consider with this prompt is their shape, their size. Are there features that stand out to you? Are there parts of their bodies that look different or interesting? What I really want to focus on is how these attributes actually facilitate their aquatic life, because it's that part of the story that helps inform our actions and management. So if these two aren't as charismatic or interesting, we can try this set. So do they have different features? What might be similar? what do we think the presence of these organisms with these particular characteristics can help tell us about their environment? And for those of you, and I expect there's quite a number of you, are involved to some degree with biomonitoring. This is the idea where we use aquatic organisms, or really any resident organisms, to tell us about the quality or the integrity or the health of their environment. So this is a often done in conjunction with other types of habitat measurements. And for those of you involved with either of our sort of stream monitoring or lake monitoring efforts, you know there's physical and chemical measurements that we take along with our biological measurements. So these biological surveys can usually be performed relatively quickly, relatively easily, and relatively cheaply. When you think about all the components that you might take out into the field, once you invest in that net, you've got it for a good long time and you can keep using it. Where chemicals need to be replaced and equipment needs to be maintained, it's fairly easy and cheap to do a biological survey. And oftentimes, we're getting unique information from the biological data. So in particular, I just want to stress two aspects, and that would be these are organisms that are living in the habitat. They're integrating changes that can occur over time. And it's not just that instantaneous snapshot that we often get with a dissolved oxygen test or a Secchi desk measurement. We're getting a time integrated perspective on that habitat. It's like asking someone who only saw the trailer of a movie what the movie was like versus someone who saw the entire movie what the movie was like. And in addition, these organisms can integrate multiple stressors. So they're simultaneously reflecting the physical and chemical environment with their presence and abundance in that habitat. So that's really hard to capture with any other sort of chemical or physical means without deploying sort of continuous monitoring sensors at every location that we would wish or hope to monitor. So that has cost and time considerations as well. But there's lots of different organisms that live in our habitats. So we might ask, well, why are we focused on invertebrates? Is that just because they're my favorite type of organism? Well, it's not a terrible reason, but probably not a good scientific reason to do this. So, in fact, many types of organisms can be used as freshwater indicators. And in some parts of our country, and indeed in other parts of the world, multiple organisms are used simultaneously for the evaluation of aquatic habitats. In general, the attributes that these organisms possess are broadly similar. So ideal indicators might have a fairly widespread distribution, which means we can expect for them to occur in the habitat. If these organisms are exceptionally rare and we don't see them in many places, then they're not as useful as an indicator. And we can also think about the diversity within a particular group. So we know that there's a lot of invertebrates, and that's what part of what makes them so interesting and fun. But other like plants and fish, they're quite diverse as well. So there's a lot of subsequent species or genera or other taxonomic levels present in the environment simultaneously. We might also be interested in how able they are to move around in their environment. Things that can just leave are probably not as informative as things that are a little more stationary or sedentary and have to sort of accommodate the conditions that they live within. And we also have their life history. How quickly can they turn over? If there are organisms that are being replaced every few weeks or months, then maybe their exposure is much more limited. If they're longer-lived organisms, then they need to integrate those changes and deal with the extremes of their environment. Then we also have some practical considerations, such as the ease of identification or the ease of sampling. Do we need a lot of specialized resources or equipment to evaluate these organisms in their habitat? And the more information we have about an organism, sort of their ecology, sort of what we know about that particular organism in the environment, can be especially helpful. And that's actually part of what I'm going to focus on here is how much do we know about a particular organism and how does that inform biomonitoring? And then I do have to mention that if you have a charismatic organism or one that's linked to tourism or recreation, like fish, they tend to get attention as well because there's some extrinsic value that those organisms bring. My husband, who studies plants, was really outraged that I didn't say that plants had extrinsic value. They obviously have value, but I don't know about a lot of botanical tourists. It's going to be a new thing. All right, so what we're here for is what do they tell us? What does the presence, what does the occurrence of these organisms in the habitat really tell us about their habitat? So what insights can they provide? What can they tell us? And what they're telling us is really through their bodies. It's through their properties or characteristics. So it's written on them and in them. And we call these features functional traits. Essentially, these are any observable, measurable characteristic that's linked to the survival and reproduction of that organism in the habitat. And some examples of this include the size of the organism attains, the type of diet that organism requires, how long that organism lives, how mobile that organism lives. All of these things can be extremely useful to us in understanding the habitat that the organism is in. Now, we do need to take a moment to consider the origin of our organisms. So I think this is particularly important. It might seem a little obscure, but I promise we'll get there. So, where did aquatic invertebrates come from? Well, most life, in fact, all life, essentially is marine, right? Everything started in the ocean, and then from there, it moved onto land or it moved into freshwater. But did it move onto land and then freshwater, or did it move directly from a marine environment into a freshwater environment? Freshwater is fundamentally different than a marine environment. The osmotic concentration is quite different, and there's many different challenges presented in a freshwater environment than a marine environment. So although they both seem like aquatic habitats and it shouldn't be that different, it's actually dramatically different. So how organisms actually made this leap is particularly important. So on your screen right now, I've got a couple examples. We've got a true bug, a giant water bug in particular. We've got a riffle beetle. We've got a mosquito. So all of these organisms actually re-entered an aquatic environment after being terrestrial. So their most recent common ancestor was terrestrial, and then they invaded freshwater. And these, in fact, still very strongly resemble their terrestrial ancestors. So that's not the case with all invertebrates. Fossil evidence is limited for freshwater invertebrates and insects in particular, but we do have some hints as to the pathway that these organisms took. So presently, evidence suggests that marine crustaceans actually moved into freshwater habitats directly. So there are marine crustaceans and there are freshwater crustaceans, and they didn't necessarily have a terrestrial intermediate between those two habitats. So that's how we get things like our freshwater shrimp and our water fleas and copepods and things like that. They had a marine ancestor, they came into freshwater. But insects, on the other hand, had a bit of a different story, or at least we believe that most insects had a different story, in that our crustacean-derived hexapods came on land, and from land went back into freshwater. And if you think this is a bit unusual, I should mention aquatic plants actually have very similar life history where they came from terrestrial ancestors as well. So this isn't that unusual or surprising. But I do need to admit, and I tell this to all of my invertebrate zoology students, that insects are, to put it simply, just crustaceans with wings. So it's just two different pathways that crustaceans took to invade more habitats. So, um, whichever pathway they took, these freshwater invertebrates went to freshwater very early on in their evolutionary history. So, some of our oldest insects are actually aquatic insects or believed to be aquatic insects. This includes some of the really charismatic fossils we have of things like the Megaisopterans or griffin flies. So these resemble modern dragonflies, and some could reach enormous sizes comparatively to our extant insects. And then we also have the Paleodictyopterans, which resemble modern day mayflies and some stoneflies. And these are also incredibly old. So, whichever way they came into freshwater, they did it very soon after that transition to land or very early on from that marine crustacean ancestor. So, those earliest insects may have been invading freshwater for a couple of different reasons. One, the concentration of different components in the atmosphere may have allowed more solar radiation to penetrate, and that could be really harmful for their tissues and cells. So, freshwater may have been a way to escape some of that damaging solar radiation because the light is attenuated by the water. And it was an exploitable habitat. There may have been less competition and more resources available, so they were able to take advantage of that. So a couple of interesting aspects for this figure. You'll notice that the two bars at the top, those are ancestral forms or extinct forms of organisms that resemble either odonates, so our dragonflies and damselflies, or mayflies. And those are some of our oldest insects, and in fact, some of our oldest freshwater insects. So that's what we mean by either that terrestrial to freshwater or marine to freshwater transition happened very early on in the evolution of all insects. And then you'll see some other groups that show up a little later. Those very clearly came from terrestrial ancestors that secondarily invaded freshwater systems to take advantage of resources and reduce some competition on land or various mechanisms. A couple of interesting things about this as well. There's good evidence, although limited, but good evidence that these earliest freshwater insects were stream-loving insects. So sorry, lake folks, the streams got insects first, probably because of the higher dissolved oxygen levels, but more on that in a moment. So with that background, that's to say that life in freshwater is challenging. It's not a seamless transition from a marine environment or a terrestrial environment. These organisms need to cope. And how I would sort of ask you to think about this is, what if we asked you to jump into a river or jump into a lake? You'd probably say, well, I need some waders, or I need a snorkel, or I need a scuba tank. I need something to help me out in that environment. And that is absolutely true, especially for the insects and other invertebrates that came from a terrestrial ancestor. They had to acquire features and adaptations that allowed them to thrive in this freshwater environment. And I'm trying to limit myself to only 5 of these topics to talk about with you. And that includes respiration, so how these organisms breathe, sort of their size and their shape and how they're interacting with that environment, their diet, how they develop in terms of their life history, and then how they move. So we're just going to take a quick look at each of these and then how that informs our biomonitoring. So let's start with how these organisms breathe. This is probably the most important biological system related to organism survival in freshwater and its use as an indicator. So first, I think it's important for me to stress that insects and other invertebrates do not breathe like we do. They don't have an active ventilation, and it's not coupled to a circulatory system like our blood. Instead, most invertebrates and insects in particular have passive systems where they're using diffusion so that oxygen can get into their tissues and their cells. So insects in particular, this is accomplished through what's called a tracheal system. This is a network of successively smaller branching tubes that have openings on the outside of the insect's body. These are called spiracles. And then oxygen just passes into those chambers, and then it gets exchanged at the cellular and tissue level, and carbon dioxide passively diffuses out. This works reasonably well in terrestrial systems, and they do need to monitor water loss, kind of like us sweating. So they can dehydrate if the trachea are open in a dry environment, but otherwise they handle it pretty well. Now, if you throw that kind of insect into a freshwater environment and it's got its spiracles open, essentially it'll drown. So they have what's called a closed tracheal system. And this is true for freshwater invertebrates and insects, as well as larval stages that might be in moist environments and parasitic forms that are inside other organisms' bodies. That's not a great place to have an open tracheal system. So here, the spiracles are closed, and oftentimes they have to compensate by expanding or increasing the surface area of those respiratory structures. So that brings us to respiration in freshwater invertebrates and insects in particular. So just like we would need a snorkel or some scuba gear to survive in a freshwater environment, these insects have evolved those mechanisms as well. So some aquatic invertebrates have really soft, thin exoskeletons, and dissolved oxygen can go right across the tissues. So just essentially passive diffusion of oxygen, because as they're using the oxygen, it's diffusing out of the water and into their cells and tissues. And that works fine for many things, especially our smallest, sort of thinnest or tube-shaped organisms. But many need to supplement the oxygen they derive from that mechanism through increased surface area, and these are gills. So gills can occur across the animal's body. We can have anal gills, we can have abdominal gills, we can have thoracic gills, so they can show up in different places. They're really useful for taxonomy. So they're also really important for the organism to breathe. So those become the sort of the best indicators that we have of water quality conditions, because they need the greatest amount of dissolved oxygen in order to survive. So they can also be indicators of water temperature, because cold water holds on to more oxygen than warm water. But then we have the cheaters, the ones with the snorkels. So we've got our larval mosquito in there as well. So we have siphons or air tubes. These are organisms that are essentially breathing atmospheric oxygen by coming up to the surface with a part of their body and then taking in atmospheric oxygen. They don't have to stay at the top. It's a bit like a whale coming up to get a breath of air, and then they can go about their life, and then they come back up periodically. So they can do that. In a similar mechanism, but without the snorkel piece, beetles and several other groups can grasp onto bubbles and use that as a physical gill, and the oxygen diffuses into the insect's body and then from the water into the bubble. And the bubble's going to shrink over time, so they'll have to replace it every once in a while, but that's how they can breathe atmospheric air underwater. So that's pretty clever. And In the most extreme environments where we have the lowest dissolved oxygen, we actually do see organisms that respire similar to us, where they kick in their circulatory system. They've got hemoglobin that can help carry oxygen throughout their tissues. So that's not something that we see across all insects all the time, but it does seem to be a feature that's really helpful in those environments. So you probably know these as the bloodworms or the blood midges if you see those in your sample. They tolerate really low dissolved oxygen conditions. And then I'm going to focus on sort of stream dwelling insects for this talk, but we could have a similar conversation about lentic habitats, so lakes and ponds. But in stream environments, we have organisms that are essentially shaped by the flow of the water in some of the most extreme ways we can imagine. So most stream-dwelling insects and invertebrates are going to be right at the substrate surface to avoid the most turbulent and highest velocity flows. But they're also going to be shaped in a particular way that facilitates their life history. So aquatic insects need to contend with drag and lift, and the water's moving over their surfaces or around them if they're hiding in the substrate. So what we see are organisms that can try different things. One approach is dorsal ventrally flattening, so flat like a pancake. These live very close to the substrate, very close to the surface, and that is in this little thin layer where the velocity is slightly lower because there's greater friction with the bed surface. So they can take advantage of the water slowing down as it's rubbing against that surface. But they can also increase their surface area so there's more contact with the surface, or they can fit into really thin little cracks and crevices. The downside is that this shape creates lift. So if they do get dislodged from the surface, they're going to tumble for a long ways. It's very hard for them to recover their position. So the other extreme is streamlining, where they become, I call it torpedo shaped, where they're really good swimmers and they can delay that drag force by being tapered in their body plan. So they can recover, they can actively go up into the water column and come back down without too much difficulty. And those that really, really want to stay put are going to have things like suckers or friction pads or hairs, or even their gills can help them to sort of stay onto the water surface or on the substrate surface. And flowing water can actually facilitate feeding in these organisms. So they'll have adaptations for obtaining nutrition from the water column, which is not something that we generally see a lot of terrestrial invertebrates doing, but something that we see a lot of freshwater invertebrates taking advantage of. And there's a lot of marine invertebrates that do this too. Filter feeding is a very popular mode of nutrition gathering. But speaking of nutrition gathering, let's talk about their diets. So this is a convenient way to sort of class our freshwater invertebrates by what they eat and how they eat it. So this can be really important for biomonitoring because it helps us to understand what food sources are available for the organisms in our site without having to measure those food sources. We see the organism and we have the evidence of their diet in their presence. So as I mentioned, filter feeding is a popular mode of feeding in aquatic environments because the food comes to you. You just have to get it out of the medium. So we have our filter feeders. We have deposit feeders, which are also taking small particles, but usually from a surface rather than in the water column directly. We have scrapers or grazers that are obtaining nutrition from that periphyton, so that matrix of bacteria, fungi, algae, that's all growing together on the matrix. It's making the rocks slippery for when we go into the stream. Piercers that are taking the contents out of plant cells, and shredders taking those larger pieces of material like the leaf fall in the autumn when all the leaves are coming off the trees. They're taking those bigger pieces and making them into smaller ones. And then predators. Got to have our good predators eating all the other invertebrates and even some of the vertebrates too. So we're going to have unique mouth parts like we see on this larval dragonfly or our black fly, which are adorable as babies, not so fond of them as adults, but they've got these filter feeding fans, or in our dragonfly, these big palps that they're using to grab their prey, so prehensile mouth part to suck in their prey. But one thing that this doesn't really capture is that diets can change with age or with the stage of life that an organism is in. So sometimes these categories miss that fact a little bit. And I'll just show you this one quick example. I collected both of these individuals at the same time at the same site when I was working on my PhD work in Canada. So at least 2 orders of magnitude size difference between these two. I don't know that they were eating the same things in the stream at the same time, so diets can shift. We have organisms that are changing size dramatically over their development. So that's our segue into development. I imagine almost all of you are familiar with the general developmental plan of insects, either incomplete metamorphosis, where we have immature stages that resemble the adults generally, except without functional wings, and complete metamorphosis, where we have organisms that don't develop their wings until after going through a rest period or a pupal stage, and then they transform into the adult form. So that's probably familiar to most of you. What I think is actually really interesting here is that these coexist in freshwater environments. It's not like, oh, all of our really older ancient groups of freshwater insects all have this incomplete metamorphosis, and they're the only ones we see in freshwater. Because we had those secondary introductions of more recent groups of insects, we actually have a mixture of both incomplete and complete metamorphosis. So instead, how I like to think about this is, are only immature stages found in an aquatic environment, or do you find multiple life stages or even all life stages in a freshwater habitat? And that's where it totally blends. So we've got both complete and incomplete representatives of each of those developments that have only their immature stages, only their juveniles in freshwater. And then we have other examples where we have complete metamorphosis or incomplete metamorphosis, where they have basically every life stage in freshwater. And that's what I think is really spectacular and interesting, because adults need to do different things than larval stages do in complete metamorphosis, but they're still finding a way to have both of those stages specialized for that habitat. And one final consideration for development is, how long does it take? So this is one of those key characteristics of freshwater organisms that makes them really valuable for biomonitoring, because they have different lengths of their life cycles. Not all insects or invertebrates develop on the same time span. Even in a temperate zone like Wisconsin, we have some organisms that have very short life cycles. We might be able to get more than one generation per year, as mosquitoes will demonstrate over and over again, or very long life cycles. And these are providing really different pieces of information to us. So we can get a short-term perspective on our habitat from our short life cycle invertebrates, and a longer-term perspective from our longer-lived invertebrates. So that combination can be really informative. And then, sort of to wrap up our life history, our functional trait piece, we can talk about the mobility of these organisms, which is also useful. So this is sort of the flip side of our size and shape. Some organisms that we mentioned are more apt to be present in the water itself and not as attached to the substrate. So like bodies, limbs are shaped for either swimming, clinging, or grasping very slippery structures under the water or near the water. And I do want to mention cases. So we have caddisflies that create cases, and these structures fill a variety of roles for the organism, including providing retreats for them and even ballast to help them stay on the bottom of the stream, and camouflage because they tend to be less mobile and more sedentary, and perhaps that's one of the reasons why caddisflies can be so informative for our analysis, because they tend to stay in that environment. It's kind of, I imagine, difficult to lug around your whole house with you all the time, great distances. So, and then the other features that we mentioned earlier, things that either help them to stay put or might help them to be more mobile. All right, so with that background, I do want to mention that not every combination of these traits occurs in nature. So we can't take one from column A and one from column B and one from column C and then find an organism that has those sets of functional traits. They are slightly more either physiologically or phylogenetically linked with other traits. So it's not an infinite number of combinations. We see some things that are more likely to occur together than others. And we can refer to these as sort of trait syndromes. We have collections of traits that seem to occur more frequently than others. And we can use these to summarize the organism. And I know some of you have had to hear me talk about this before, but I imagine these list of attributes a bit like the stats on a baseball card or a hockey card, or as I'm told by my students, a Pokemon card. So that's what I'm showing here, sort of our homemade version of this trait card for a macroinvertebrate that's used for this type of monitoring. So we try and summarize this information for these organisms. All right, so how do we put this together? How do we put together a biotic index? So as I mentioned, many of you have probably participated or have been involved in biomonitoring and have contributed data and or calculated a biotic index. So it summarizes our biological community. It helps us to interpret ecosystem quality or integrity or health. And it helps to communicate that information to other people, the people that live in that area, near that habitat, or in that watershed, near that lake, and other stakeholders, managers, government officials, etc. It helps us to track things over time and with management activities to see, did that change make a positive impact or not, and compare locations, sites, habitats, or impacts. So I'm hoping that many of you are actually familiar with this. Our WAVE, our Water Action Volunteers, volunteer stream monitoring. So this is one instance of a biotic index. And here we're focusing mostly on presence absence of these organisms. We're not completely concerned with how many of them we're seeing, so we don't need to consider abundance. But what I want you to notice, and I'm sure you have, is that these are divided into 4 groups. If you've ever wondered why a particular organism is in a particular group, I'm hoping to answer that a little bit today. So it really comes down to those functional traits and the sensitivity that those trait syndromes provide for those organisms. So our group 1 organisms, these are the less tolerant or perhaps least tolerant of the organisms that are in our biotic index. They rely on dissolved oxygen almost exclusively, so either cuticular, so across their exoskeleton, or with the aid of gills. That means they're really restricted to cool and cold water habitats, and they tend to be long-lived, and they tend to be predators. So predators, we have fewer of them in the environment. You need lots of prey to feed a fewer number of predators, so we tend to see fewer of these. So that's sort of our group 1 category. Group 2 is very diverse. There's a lot of organisms in group 2. My cards here, there's more of them than you probably see on the waveform, and that's because we can have multiple taxonomic groups that represent each of those pictures. So that's why there's so many cards for this group. These are slightly more tolerant or a little less picky, is what I'd say. Most still rely on cuticular respiration or gills, but our body shape and size starts to vary. We have some really, really big guys, crayfish and freshwater mussels, but we also start to see some more smaller sized organisms as well. We also have a wider range now of functional feeding groups. Things like our collectors show up, some of our scrapers show up in this group. And now most of them have annual life cycles. So that gives you an idea. So slightly shorter lived, except for maybe our crayfish and our freshwater mussels. Group 3, these are becoming even more tolerant. We have those body shapes that really facilitate oxygen transfer, so they can live in environments that contain less dissolved oxygen. Again, we really see a preponderance of annual life cycles and a switch in our diets to mainly collectors and grazers. So eating plant matter or that biofilm, the periphyton that's at the bottom of our streams and our lakes, or the small bits of detritus that are floating and then sinking to the surface. And then finally, we have our most tolerant group. And here we start to see some of the most dramatic adaptations for respiration. So some of our snails actually have a structure that can act like an air sac or a functional lung. So they can live in particularly low oxygen environments. And we see groups that can possess that hemoglobin that really integrates the circulatory system with the respiratory system to compensate for low dissolved oxygen in those environments. And again, here we see an absence of predators, mainly collectors and grazers in this group. So those sorts of insights are what's provided from this biotic index. We can see that the presence of these organisms, and you'll probably always have group 4s, you'll probably always have group 3s, but the higher numbers of taxa within groups 2 and 1 can really help us to understand the relative health of our ecosystem. So we put this together in our calculation where we take into account the numbers of organisms, or the numbers of types of organisms, I should say. We're not strictly concerned with abundance here. And then we get our index score. So this is really giving us a single numeric value that can be associated with our quality status or rating, and this facilitates those spatial and temporal comparisons. So if we're looking across to different areas or across time, this provides a mechanism for us to communicate that. What some of you may be surprised to learn, or maybe not surprised to learn, is Wisconsin is not the only place that does this. And I think it's interesting and informative to take a peek at what some other groups do for their biotic index. So same concept, same basic scientific underpinning, but different versions. So when I was a master's student in Iowa, we had Iowa water, and this was the index we used for that. I don't know that Iowa water is still going on, but this is what we did back a few years ago when I was a student there. And you'll see that here, instead of 4 groups, we have 3 groups, and it's the number of species or number of taxonomic units within each of those groups that's measured. So again, not using abundance, but using the diversity of those organisms to help inform that index. So it looks a little different, functions a little different, but it has that same sort of basis in the functional traits of those organisms reflecting a status of our stream. So that's a different geographic location. We can consider sort of a different habitat or a different sampling mechanism. So we could think about the organisms that are maybe restricted to leaf packs. So there's an index specific for examining the organisms associated with leaf packs. Again, here we have 3 groups instead of 2. One of the things that my students get thrown off by is they do ask for the sum of individuals, even though it is not used in the calculation for the index. They all get stuck here, but that's okay. So we're counting the number of taxa within each of our groups, and then scaling that for our interpretation. And you'll see one thing that's interesting about this one is they're separating sort of other caddisflies from our net spinning caddisflies. So that's a differentiation that we're making based on life history characteristics of that group. Now to go sort of across the world, we can take an example from the UK and their Riverfly Partnership Program, where they look at only eight, only eight organisms, and they are concerned with abundance. So they actually try and scale how many of each of these groups are present. I guess if you're only focusing on eight, it's a little easier to count them in your process. And they're putting that information together in their index as well. Go back to North America, but sort of our neighbors to the north. We have the Ontario Stream Assessment Protocol. Looks a bit more like WAVE, so maybe a little more familiar, similar pictures. And they're looking at, again, the abundance of these organisms in particular groups as well. So now you might be wondering, well, which one is right? And how do we know if they're right? And maybe how can we make them better? Because there seems to be some variability depending on whether we count taxa or count individuals. All of these represent community science biotic indices that tend to be simplifications of much more detailed and laborious either multivariate or multi-metric analyses that regulatory agencies, either for the relevant state or province or federal entity, is really interested in. But in general, the citizen or community science biotic index should generally follow what we can calculate with those more sort of laborious laboratory-based analyses. So again, just focusing on Wisconsin, although it is used in many, many other jurisdictions, including Canada, we have the Hilsenhoff Biotic Index. This is one of the earliest and still very much in use biotic indices. This one's the family level version. There are other versions that are much more detailed. And this was developed by a Wisconsin entomologist named William Hilsenhoff. And here we take the tolerance value for the organism. So take essentially the sum or the weighted average of all those functional traits for that organism. And that becomes its tolerance value, a bit like the group that it belongs to on the waveform. And that is integrated into this index that takes into account how many of those specimens were observed in the sample. So we have their tolerance value, we have their abundance, and we have the total abundance for the sample. And that gives us a biotic index value with a water quality rating. And we can compare how our work matches sort of this laboratory-based measure. So that's one way that we can make sure that our community science measures are living up to our expectations. So some of you may have noticed that throughout this presentation, I have not given a lot of scientific names. There's a couple reasons for that. One, I get asked to do a lot of talks about taxonomy, so it's nice to take a little bit of a break from that. And two, I want to talk about specifically what is the value of taxonomy. So taxonomy really is a way for us to provide a shorthand notation to all of that ecological information about the functional traits, abilities, and attributes of the organism. It's a bit like me trying to explain to you the plot of a movie that I can't remember the title to. I can list off a bunch of characteristics, and you may have an idea of what I'm talking about, but you may not be able to pinpoint the exact organism I'm talking about. But if I give you the name, now we have a shared understanding about what that organism is and does in the stream or the lake environment. So taxonomy is critically important. I don't want to underemphasize that, but I do want to sort of overemphasize that it's what these organisms do that's really important for the biotic index. But the taxonomy here links the label, i.e. the name, to that natural history information, and it also enables us to have a conversation about those organisms in a meaningful way. So we know we're talking about the same organism, and that can be incredibly important. So to put all of this together, we've demonstrated that these biotic indices differ across jurisdictions for different purposes or study objectives, but they're all based on the same scientific principles. We have organisms that have adapted over evolutionary time to particular considerations and properties of their environment, and we can use that to infer the quality of the habitat for our purposes. But that doesn't mean that every biotic index is perfect. And it doesn't mean that they don't need to be examined critically from time to time. So there's ways that we can refine or improve biotic indices. And you can see from the variety, just the small sampling that I've provided here, that there's different approaches or ideas on how to do that. And this may involve increasing the resolution in the organisms, so understanding to a greater degree the sensitivity or the response to particular stressors. So really understanding if we summarize something into a particular group or we give it a specific tolerance value, what does that mean and how is it linked? Do these change over time? Do they change with life stage? I think that's really interesting. And then there's the mechanics of the index itself. So do we include abundance? You know, that's more time consuming in the field, but is it inherent and valuable from the index perspective? Or do we just need to change how it's calculated? Do we need to change the factors that we multiply for each of the groups of organisms? And how do we put that score together to better match what we think is the condition on the ground? So I do want to mention that even the more rigorous lab-based measurements are still essentially estimates. All of these are imperfect, but we want to have the best estimates possible. It's a bit like statistics. You know, we know very few things with absolute certainty, but if we have a higher degree of certainty about something, we think that's closer to biological truth or meaning. So perhaps we can tweak that calculation or the mathematical components of that index. And we can look at different data sources. So as we expand our knowledge of aquatic invertebrates, we can add more traits and think about how our information about those traits can change a position or an association with a group or a tolerance score. So there's still lots for us to learn about aquatic organisms and what they are in fact telling us about that stream environment. So I know this has sort of been a bit of a whirlwind tour. Hopefully there was some new information for you. And I'd be happy to take any questions. Thank you so much for your attention today.
Thank you, Jessica. That was awesome. Our first question is regarding your functionality cards that you have. And the person's wondering if they can get a set of those cards, especially the ones that are on their biotic index sheet.
Yes, these are first edition biotic index trading cards, and I have PDFs of those. And I should say that currently we only have WAVE individuals as those cards, but if folks are interested, I know my students and I don't need much persuasion to try and make some more because the list of taxa for WAVE is actually pretty short. There's basically no true bugs on there and only a small number of beetles. So there's actually a lot of other fun guys that I'd love to make cards for. I just need an excuse. So yes, you can reach out to me. My email's on the screen and I can send those your way if you're interested. Or maybe there's another mechanism that Katie or Emily or Paul can share to distribute those to folks. But I'm happy to share our trading cards.
Great. The next question is, are the more tolerant organisms, groups 3 and 4, also present where you find the less tolerant organisms in groups 1 and 2?
So I don't have a lot of data to back this up, just some ecological understanding. I think it's impossible to find groups 1 and 2 without also seeing some groups 3 and 4. I don't think they're ubiquitous, and just because they're tolerant does not mean that they're not going to show up in a healthier habitat. They're basically everywhere. So essentially groups, especially group 4 and most in group 3, are essentially ubiquitous. You're going to see them everywhere. It's some members of group 2 and especially group 1 that are going to be the ones that are absent from those habitats. I don't know if I fully answered that question. So Paul, let me know if I missed something there.
Yep, I think you did. The next question is about neonic pesticides, neonicotinoids. The person is asking regarding less and less stoneflies and mayflies in the last couple of decades. Is there anything that you or your group is doing to address the threat of neonicotinoid pesticides, or is there anything that TU chapters or the public could assist with to reduce the use of these pesticides?
That's an excellent question. I'm not an expert in ecotoxicology. I have a broad understanding that these impact organisms that have longer life cycles. They have less ability to exit the stream or exit the environment than other insects do. So that's why they seem to be disproportionately affecting some of those really sensitive taxa. So that's our mayflies, our stoneflies, and some of our caddisflies as well. I don't have specific strategies related to neonics, but I think by informing the public about how biotic indices work and what are the inferences we can make from biotic indices, as well as some of the work that my students are doing to try and sort of tweak or modify biotic indices to make them more informative, can help us to make positive changes for stream management, including trying to reduce pesticide use, especially those neonics. So sort of a hedgy answer because we're not specifically focused on that, but I think there are parallel aims that the better we can inform the public about how these operate and get more people involved in participating in these efforts, we can help to address that concern.
Okay, the next question is the Hilsenhoff slide listed organic pollution as the indexing stressor, and you mentioned DO as an essential factor. Could you elaborate on the tolerance and classification levels in relation to various stressors?
Yes, so a main, which is, I'll back up. Why I started focusing on respiration is because that's really the key to some of our first work on biotic indices. So as debris accumulates in bodies of water, bacteria are breaking it down, and that's an oxygen consuming process. So even if it looks to us like that habitat is fine, underneath the surface, there could be depleted oxygen, and that's one of the first things that's going to prevent an organism from being successful and surviving in that habitat. So some of our most fundamental metrics and index values for freshwater systems are really linked to the respiratory mechanism of the insect and the oxygen environment of the habitat. So for example, you can think of a similar application with fish, where there are different indices for cool water, warm water, and cold water streams for fish, because they also have different oxygen requirements. So generally, the tolerance value is correlated with that feature, but it's sort of like a partial correlation because there are things that can modify that. So that's where we see our most extreme sensitive groups also being long-lived predators that also have those added sensitivities. So you could do a tolerance value across the board just on respiratory mechanism, but then we can augment it a bit with some of those other traits. And that's still sort of the state of the science right now, is taking into account the oxygen tolerance and then augmenting that a bit for sensitivity to other aspects. So the concomitant effect of stream temperature or other stressors. So I would love to say that we have an index for some of those pesticides or pharmaceuticals, but we don't have enough information about a broad enough set of organisms to create tolerance values specific to those stressors yet.
All right, we have one quick comment left and then one more question. So that'll wrap us up here at about 1 o'clock. The comment is just mentioning that there's an app called Pocket Macros. That's a helpful app for identification, and it's set up similar to the Wave sheet. And the other question was looking for more clarity on how bloodworms combine circulatory and respiratory systems.
Yes, so they're not combined necessarily, but because of the proximity in the tissues of the organism, they can diffuse oxygen into essentially their blood cells. So those are hemocytes within the insect, and they actually have hemoglobin, which is essentially the same as the hemoglobin that's in our red blood cells. So it grasps the oxygen molecule and then it passes through the body of the organism with the circulatory system of the critter. And then once it's used, it exchanges it with dissolved carbon dioxide to swap places. So they're not quite as intimately connected as our respiratory and circulatory system. It's just the proximity of the tissues being close to one another allowed for the oxygen to transfer to that hemoglobin molecule and move through the tissues of the organism.
Wonderful. That wraps up our Q&A.
All right. Well, thank you again so much for inviting me to your Winter Water Talk series.
Thank you so much, Jessica, and thank you everyone who joined us today. We had a big group and a lot of participation, and we'll be working on getting the recording uploaded to YouTube so everyone can watch it over and over and share it with their friends and family. So everyone take care, and thank you again.